Hey guys, welcome back to One Stop Biology. So guys, in today's video, we are going to understand what is the C4 pathway. So let me remind you that we are right now dealing with the photosynthesis in higher plants chapter, which is chapter 13 of class 11th. And we have already covered till the Kelvin cycle. So in the last video, we actually covered the Kelvin cycle. And guys, I hope you are going through all the videos because now the board exams are very close, and this is all about the NCERT series for boards and NEET. So please do let me know if you have any questions, if you have any doubts in regarding the videos, or if you want me to improve, if you have any particular feedback regarding the videos. And yes, guys, if you like the videos and if you're going through the videos, please do like it and share it with all your friends. And if you're new to the channel, please do subscribe to the channel and press the bell icon to get all the notifications. So guys, let's start with the C4 pathway. Now, in the last video, we also saw that the first, right, the first product which is formed, right, in the C3 pathway is what? It is phosphoglyceric acid, 3-PGA, and that is why the Calvin cycle is also known as C3 pathway. In similar way, C4 pathway is named after the first product which is formed in this pathway, which is the 4-carbon oxaloacetic acid, right? So because of that, the pathway name is C4, and the plants in which you will find C4 pathway are known as C4 plants. Now, how they are different and how the C3 and C4 pathway is different, we'll see that as well, right? So, guys, there are certain plants which are basically adapted to dry tropical regions, right? There are plants which are present in the dry tropical regions, and these plants adapt to C4 pathway, right? But that doesn't mean that they do not have C3 pathway, right? So, though these plants have C4 pathway, C4 oxaloacetic acid, they do make it. They use C3 pathway or the Calvin cycle as the main biosynthetic pathway. Right. Then let's see what is the difference exactly. So C4 plants are special, guys. We say C4 plants are special. Now, how are they special exactly? So they have basically special type of leaf anatomy. They can tolerate high temperature, right? So I'm saying they have special type of leaf anatomy. They can tolerate higher temperature. They show response to high light intensities, and they lack photorespiration, and they have higher productivity of biomass, right? So basically, the C4 plants have large cells around the vascular bundles. They have large cells around the vascular bundles. And these cells are known as bundle sheath cells, right? And these leaves which have bundle sheath cells, right? They have a typical anatomy, right? And this anatomy is known as Kranz anatomy. And here, this word Kranz means wreath, right? And this is a reflection of arrangement of cells. Okay, so these bundle sheath cells may form several layers around the vascular bundles, and they are characterized to have large number of chloroplasts. They have large number of chloroplasts, right? Now, guys, they have thick walls. They also have thick walls, which is basically impervious to any kind of gaseous exchange, and they have no intercellular spaces. They have no intercellular spaces as well. Okay, so these bundle sheath cells, they have large number of chloroplasts, they have thick walls, and they have no intercellular spaces. So if you basically cut a section of a C4 plant like a maize or a sorghum plant, right, you can observe this Kranz anatomy and you can also observe the distribution of mesophyll cells, right? Now, if you will observe these leaves under the microscope and if you look for the bundle sheath cells around the vascular bundles, right, you can identify these C4 plants very easily, right? And there is a specific kind of pathway in these plants, right, which is again a cyclic process, and this pathway is named, right, Hatch and Slack. This pathway is also known as Hatch and Slack pathway. Okay, this is named after the scientists, after the biologists. Now, what happens here, right? Let's see what happens here. So, if we see what are the primary acceptors, first, what is the primary carbon dioxide acceptor? Here, the primary carbon dioxide acceptor is PEP, which is phosphoenol pyruvate. So this is the primary acceptor, and the enzyme responsible here is PEP carboxylase. PEP carboxylase, or we call it PEPcase. Right? Now, why this is the primary acceptor and why not the Rubisco enzyme or why not the ribulose bisphosphate? Because here the C4 cycle is happening in the mesophyll cell and bundle sheath cell, and mesophyll cells lack Rubisco enzyme, right? So instead of the C3 product, here they form a C4 acid in the mesophyll cell first, right? So first it forms the phosphoenol pyruvate, from that it forms C4 acid, and then it forms a 4 carbon compound, the C4 acid, which is either malic acid or aspartic acid in the mesophyll cells, and these C4 acids are transported to the bundle sheath cell. Now, what happens in the bundle sheath cell? So, in the bundle sheath cell, the C4 acids are broken down to carbon dioxide and a C3 molecule, right? So, in Kelvin cycle, the C3 molecule was being formed directly. Here, it is getting formed from C4 to C3, right? So now we have the C3 molecule, which is again transported back to the mesophyll where it is converted to the first product, the primary carbon dioxide acceptor, again, which is phosphoenol pyruvate. Now, here what happens is that one is carbon dioxide release. So, when it is transferred to the bundle sheath cell and when decarboxylation occurs and when the C4 changes to C3, there is release of CO2. So there is a release of CO2 in the bundle sheath cell, and this CO2 enters the C3 cycle, right? This enters to the Kelvin pathway. Okay. Now these bundle sheath cells, remember guys, these bundle sheath cells are rich in the enzyme Rubisco, right? But they lack the other one, which is PEP carboxylase or PEPcase, right? So basically here, after the carbon dioxide is released, the Kelvin cycle happens in the bundle sheath cell. So basically now Kelvin pathway is common. We know that this Kelvin pathway is common to C3 and C4 plants, right? Now, Kelvin pathway occurs in mesophyll cells of C3 plants, but in C4 plants, it does not takes place in Remember, guys, it is very important. In C4 plants, it does not takes place in the mesophyll cells because they lack the Rubisco enzyme. Instead, it takes place in the bundle sheath cells, right? So if you even want to see this cycle in the form of this flow chart, let's see this same cycle in the form of a flow chart. Okay, so basically, guys, here the first cell that you see, which is in green color, completely in green color, is the mesophyll cell. Okay, so this one is the mesophyll cell, and the gray one is the bundle sheath cell. Okay, now here again, atmospheric carbon dioxide is coming inside, right? So basically, this is the plasma membrane, and this outer one is the cell wall, right? And this connection between the two cells is plasmodesmata. Okay, now what is happening is the atmospheric carbon dioxide is coming inside the mesophyll cells, and now you have the carbonate ion and phosphoenol pyruvate, right? So the phosphoenol pyruvate here is what? It is the primary carbon dioxide acceptor. Now this fixes phosphoenol pyruvate fixes carbon dioxide and it gets converted to a C4 acid. This process is by fixation. So this is known as fixation. Then this C4 acid is transported to the bundle sheath cells. So this C4 acid is transported to bundle sheath cells. Now here we have decarboxylation happening. So because of this decarboxylation, the C4 acid changes into C3 acid and we have carbon dioxide released which is fixed into Kelvin cycle. And now here in bundle sheath cell, the Kelvin cycle is happening. And this C3 acid is again transported to the mesophyll cells, right? And it is now regenerated, right? So regeneration happens and it is regenerated into the first carbon dioxide acceptor, the primary carbon dioxide acceptor, which is phosphoenol pyruvate. And this cycle keeps on repeating. So here what is happening is since mesophyll cells, these cells lack Rubisco enzyme, the C3 cycle is happening here. C3 cycle is happening in the bundle sheath cells, not in the mesophyll cells, right? So guys, I hope you would have understood the entire concept of C4 cycle and why it happens in the C4 plants, right? What exactly these plants are and how they are different from the normal C3 plants. So please do like the video and share it with your friends. And if you have any doubt, guys, please do not hesitate in asking your doubts. You can just mention your questions in the comment section, or you can also send in your questions on WhatsApp. WhatsApp number is given in the description of the video. And yes, guys, if you are new to the channel, please do subscribe to the channel and press the bell icon to get all the notifications. Thanks for watching this video, guys. See you in the next one. Take care. Bye bye.